Dear students, in today's lecture, we will learn about autophagy. Let us start with the introduction. Autophagy is defined as the catabolic process that digests cellular contents such as cytoplasm, malformed proteins, long-lived proteins, and damaged organelles inside lysosomes. Autophagy is accelerated by various cellular stressors such as nutrient starvation, DNA damage, and organelle damage, and it protects the cell by facilitating the degradation of superfluous or damaged cellular constituents. Although sometimes hyperactivation of autophagy can lead to cell death. Autophagy allows degradation of macromolecules such as proteins and organelles and provides a source of monomolecules such as amino acids, fatty acids, and nucleotides for reuse in the cell. Let us now take up the types of autophagy. There are three main types of autophagy, that is macroautophagy, microautophagy, and chaperone-mediated autophagy. Let us take up the first type, that is macroautophagy. In this type of autophagy, a part of a membrane structure forms an isolation membrane or phagophore by a process called nucleation, which elongates, expands, and terminates to form a double membrane bound vesicle containing the cytoplasmic components, referred to as an autophagosome. The autophagosomes then transports the contents and fuses with the lysosome to form an autolysosome, where the contents are digested by lysosomal enzymes. The term autophagy usually indicates macroautophagy unless otherwise specified. The next is microautophagy. In microautophagy, the cytosolic components are not delivered through an intermediary vesicle but are directly taken up by the lysosome by invagination of the lysosomal membrane. The components are then degraded inside the lysosomes. Both macro and microautophagy vesicles can engulf large cellular structures through both selective and non-selective mechanisms. The third one is the chaperone-mediated autophagy. Chaperone-mediated autophagy is different from the other types of autophagy in two ways. First, it carries out selective degradation of cytosolic proteins, which are usually labeled with chaperone-mediated autophagy target motif. And secondly, the mechanism of delivery of the targeted proteins to lysosomes is different. In this method, only the targeted proteins with targeting motif in their amino acid sequence is recognized by heat shock cognate protein of 70 kilodalton, known as HSC17, are translocated across the lysosomal membrane in complex with the chaperone proteins. The substrate and chaperone complex is recognized by the lysosomal membrane receptor lysosomal associated membrane protein 2A or LAMP2A, which is a receptor for chaperone-mediated autophagy. After binding to the lysosomal membrane, the targeted proteins unfolds and crosses the lysosomal membrane with the help of a lysosomal form of HSC70 and enter the lumen where they are degraded. Next, let us take up mechanism of autophagy. The formation of the double membrane vesicle during autophagy is a complex process involving 16 conserved autophagy-related proteins or ATG proteins and 2 other ubiquitin-like conjugation systems. Most of the autophagy-related proteins or ATG proteins were identified in yeast. The role of the ATG proteins and conjugation systems in autophagy may be summarized as follows. The first process is induction of autophagy. The next is autophagosome formation. The next step is the degradation. Next is the reuse. Let us take up induction of autophagy. The most typical trigger of autophagy is nutrient starvation, and lack of any essential nutrient can induce autophagy. For example, nitrogen starvation is the most potent stimulus in yeast, while nitrogen or carbon starvation triggers autophagy in plant cells. In case of mammals, regulation of autophagy is complicated and is controlled by many factors. The next is autophagosome formation. Autophagosome formation generally starts with sequestration of cytoplasmic constituents including organelles by a unique membrane called the phagophore or isolation membrane, which is like a Golgi cisterna. The complete sequestration of the elongating phagophore results in formation of the autophagosome. During the initiation of autophagosome formation, 2 complexes are required. The first one is a complex that contains the class 3 P13K VPS34, then ATG6 oblique BECLIN1 ATG14 and VPS15 oblique P150.73N. Another complex with a serine or threonine kinase ATG1, which requires 2 other autophagy proteins, that is ATG13 or ATG8 and ATG17 for the kinase activity of ATG1. The elongation step involves 2 ubiquitin-like conjugation pathways. The ATG8 or MAPLC3 microtubule associated protein like chain 3 or GABARAP or gamma aminobutyric acid type A receptor associated protein oblique GATE16 or Golgi associated ATPase enhancer of 16 kilodalton and ATG12 systems. During autophagosome formation, the cysteine protease activity of ATG4 cleaves the carboxyl terminal of the soluble ATG8 protein to expose a reactive glycan residue. ATG4 requires ATG7, which is an E1-like, and ATG3, which is E2-like enzymes for its activation, and further, the function of ATG3 requires a protein complex of ATG5, ATG12 and ATG16. The activated ATG8 or ATG8-PE in yeast and lipidated LC32 in mammals then binds covalently to phosphatidyl ethanolamine, which remains bound to the autophagosome membrane until some of it was cleaved by ATG4 to be recycled or may get degraded within the autolysosome if unclipped by ATG4. The completion of the autophagosome formation involves the dissociation of the ATG16, ATG5, ATG12 complex from its bordering membrane, and the components are recycled by ATG2, ATG9 and ATG18. The completed autophagosome is now ready for fusion with the endosome or the lysosome. The next step is the degradation. After the fusion of the autophagosomes with the lysosome or in metazoan cells or vacuoles in yeast and plant cells, the inner membranes of the autophagosome and the cytoplasmic contents in the autophagosomes are degraded by lysosomal or vacuolar hydrolysis. Such degrading structures are commonly called autolysosomes or autophagolysosomes. Next is the reuse. The monomeric units, commonly amino acids, left after the degradation of macromolecules in the lysosome or vacuoles are exported to the cytosol for reuse. Next, let us study about regulation of autophagy. Autophagy is generally activated in response to different stress and physiological conditions such as food deprivation, hypothermia, and hypoxia. These stresses and physiological conditions are known as major environmental modulators of aging and are also the conditions that induce autophagy. The molecular pathways of autophagy also displays many interrelations with factors that influence aging. Some of the factors that regulate autophagy can be discussed as follows. The first one is regulation of autophagy by insulin or insulin-like growth factor 1 or IGF-1. Insulin or IGF-1 or transforming growth factor signaling pathways has been reported to mediate the influence of certain autophagy genes on cell growth in many organisms including mammals. The disruption in the function of autophagy genes also inhibits morphogenesis and survival in many organisms. The second one is regulation of autophagy by mammalian target of rapamycin or mTOR. Treatment with rapamycin induces autophagy in many organisms and the mechanism is regulated by mTOR in mammals. It regulates the induction of autophagy by controlling the activation of several effectors that regulate transcription or translation of certain proteins required for autophagy through phosphorylation. The mammalian target of rapamycin or mTOR acts as a negative regulator and regulates autophagy in cooperation with other proteins upstream or downstream of mTOR signaling. The next is regulation of autophagy by damage-regulated autophagy modulator and p53. p53 has been shown to play a role in damage-induced apoptosis by a damage-regulated autophagy modulator dependent mechanism causing full cell death in human cancer cell lines. Damage-regulated autophagy modulator is a integral protein of the lysosomal membrane and it is a direct target of p53 which induces macroautophagy and helps in accumulation of autophagosomes. p53 suppressor has been shown to be mutated in many forms of human cancers resulting in induction of autophagy. The fourth is the regulation of autophagy by forked head box-O or FOXO and reactive oxygen species or ROS. Recently, activated FOXO has been shown to increase the level of transcription of several autophagy-related genes that induce protein degradation during autophagy. Similarly, reactive oxygen species also regulate starvation-induced autophagy. When reactive oxygen species accumulate during various cellular activities causing oxidative stress, reactive oxygen species may act as signaling molecules and induce the autophagic process. Let us now take up the functions of autophagy. The first function is utilization of degradation products. During starvation for several hours, degradation of proteins by autophagy is upregulated as an adaptive response and provides the necessary amino acids to the cell. Excess production of amino acids by induction of autophagy is an acute response or emergency action and can support cell survival only for a short time. The second one is elimination of cytoplasmic contents. Autophagy plays important role in intracellular elimination of proteins or cellular organelles by digesting or degrading them. This role of autophagy is critical for the cell or organism, and defects may cause various cellular malfunctions including neurodegeneration and tumorigenesis. Many human neurodegenerative diseases including Alzheimer's disease, polyglutamin or CAG repeat diseases and Parkinson's diseases has been shown to be associated with accumulation of autophagic vacuoles. In tumorigenesis, autophagy may serve as a mechanism for survival in tumor cells in many cases, but other reports suggest that autophagy may also act as a tumor suppressor. The third function is the transportation from the cytoplasm to the lysosome or endosome. Autophagy can provide a pathway to the cells for the transport of molecules from the cytosol to the lysosome or vacuoles. For example, the transport of vacuolar enzymes or APE1 and AMS1, which are synthesized without signal sequences in the cytosol, are transported to the vacuole by the autophagy-related cytosol pathway in yeast. The endogenous antigens on major histocompatibility complex or MHC class II molecules are also presented on the cell surface by autophagy. The next function is sequestration or packing. Sequestration in autophagic membranes without involving degradation may also occur in response to several stresses. For example, during ER stress in both yeast and mammals, ER containing autophagosomes or ERAs, which do not fuse with vacuoles, are formed in the cytoplasm and plays a protective role against cell death. Although autophagy acts as a defense mechanism against some viruses, some pathogenic organisms may establish intracellular niches for survival and replication in autophagic or related vacuoles by subverting the autophagic pathway. Next is autophagy and cancer. The role of autophagy in cancer may vary depending on the context. Autophagy may suppress tumorigenesis in some cancers, while it may enhance tumorigenesis and protects tumor cells from cell death in other cases. Autophagy may serve a protective role, allowing cells to survive during nutrient deprivation, and when autophagy is prevented under these stressful conditions, cells undergo apoptosis. However, when tumor cells are starved, autophagy may inhibit apoptosis and prevent them from dying and enhancing tumorigenesis. On the other hand, autophagy may serve as a tumor suppressor and play an anti-cancer role by several mechanisms, including damage caused by reactive oxygen species by removal of damaged mitochondria and other organelles, or by limiting tumor cell growth, reducing mutagenesis, or by killing developing tumor cells. The next function is the role of autophagy in cell death. Autophagic or type 2 cell death may contribute to programmed cell death in addition to apoptotic or type 1 cell death, although the pathway is less understood compared to the molecular pathways for the regulation and execution of apoptosis. The next function is the role of autophagy in diseases. In addition to its role in cancer, autophagy also plays important role in numerous diseases including bacterial and viral infections, neurodegenerative disorders, several myopathies like Danon disease and Pompe diseases, and cardiovascular diseases. Let us now see the conclusion. Autophagy is an evolutionary conserved genetically programmed process for degradation of long-lived cellular proteins and organelles in lysosomes. Autophagy plays an important role in development and is commonly induced in response to changing environmental stimuli. Autophagy may serve a protective role, allowing cells to survive during nutrient deprivation, and when autophagy is prevented under these stressful conditions, cells undergo apoptosis. When tumor cells are starved, autophagy stops them from dying by inhibiting apoptosis. However, autophagy may also suppress tumor growth in some cancers, and it makes the role of autophagy in cancer complicated. In addition to its role in cancer, it is also important in numerous diseases including bacterial and viral infections, neurodegenerative disorders, several myopathies, and cardiovascular diseases. Thank you.